Hi, my name is Tyra Fong, and this is my presentation for Elements of Effective Communications. Since 2014, the world has been on high alert about the outbreak of the Ebola virus disease from Africa. This virus is notorious in how it affects its victims. Healthcare workers are continuously learning how to protect themselves and others from Ebola. There are many different ways that the world can help control the Ebola virus disease so it does not spread any more than it is. Researchers have been working hard to understand how the Ebola virus disease is transmitted, how to diagnose the virus, and how to treat the virus. Being informed about the Ebola virus disease will help people so they will not contract this disease. The Ebola virus disease is a single-stranded, unsegmented RNA virus that has a filamentous structure. This virus belongs to a family called Filoviridae, and filoviruses are transmitted to humans from animals. They are also known as a zoonotic virus. Ebola is an etiology of a viral hemorrhagic fever, and a syndrome that is complex and characterized by fever, capillary bleeding, and shock. An etiology is the study of causes and usually used in medicine. It is the science that deals with the causes or origin of disease. Within the Filoviridae family, there are 5 different strains that have been identified and isolated: Typhus, Sudan, Zaire, Russian, and WGU. The Ebola virus disease is believed to be transmitted from animals. There is evidence, though, that fruit bats are a reservoir for spreading this disease. There are 3 species of the fruit bats that are carriers for the Ebola virus disease: the hammer-headed fruit bat, Frankfurt epaulette fruit bat, and the little collared bat. Of the infected animals that were tested, immunoglobulin G specific for Ebola virus was detected in serum within these 3 species of fruit bats. Other animals besides fruit bats that can transmit Ebola are gorillas, chimpanzees, forest antelopes, and porcupines. There is a higher mortality rate among the great ape species during dry seasons because fruits are scarce during this time, so they feed more on bats. Humans contract the Ebola virus from poorly cooked infected animals, especially chimpanzees, because they feed on bats, which could have the disease already in their system. Locals that live in outbreak regions should be educated better so they can help and protect themselves from contracting and spreading the Ebola virus disease. Since Ebola is a zoonotic virus, animals first transmit the disease to humans by blood secretion or bodily fluids. Once the human is infected, it takes 2 to 21 days for the virus to incubate. A person can transmit the virus to another person easily by close personal contact with infected individual through blood, secretion, and bodily fluids. Surfaces and materials that are contaminated with the infected person's bodily fluids is another way a healthy person can contract this virus. That is why healthcare workers are always at risk for contracting Ebola when they are caring for Ebola-infected patients. It is important that healthcare workers understand how to use personal protective equipment, and the equipment and training are always available for hospital workers. A personal protective equipment includes a face shield, a non-sterile long-sleeve gown, goggles, and gloves. Another way Ebola can be transmitted from person to person is through the deceased. Burial ceremonies where mourners have direct contact with the body of the deceased person do play a role in the transmission of the Ebola virus disease. When a person is positive for Ebola, they remain infectious as long as their blood and secretion contain the Ebola virus. A person does not transmit the virus during the 21-day incubation period, but they do become infectious once they develop symptoms of Ebola. There are particular tests that are needed in order to confirm a patient has Ebola. Initially, the Ebola patient will have symptoms just like the flu. By the 3rd day of being infected with Ebola, the patient will have hemorrhagic manifestations and bleeding, which are externally and internally. Lastly, by the 1st week, a patient will have a transient measles-like skin rash. Once a person thinks they are infected with Ebola, they should get a lab testing a few days after symptoms start to show. The lab findings will show that the patient will have low white blood cells and platelet counts with elevated liver enzymes. Specific investigation is used to confirm the Ebola virus disease because it is difficult to distinguish EBD from other infectious diseases. In the lab testing, technicians can use a PCR or polymerase chain reaction and a virus isolation by cell culture to determine whether a person actually has Ebola or has died from the virus. They are known to be asymptomatic. There are chances that a patient can recover from being infected with Ebola. They are known to be asymptomatic, meaning they have no symptoms. A patient is considered asymptomatic if they have recovered from an illness or condition and no longer have symptoms. When an infected person recovers from Ebola, they have these antibodies that they develop, and they last for approximately 10 years. A side effect, though, is that a recovered patient might have long-term joint pains and problems with their vision. It is still unknown if the Zaire strain can be infected with a different strain from the same family. Health officials have found some treatments for the Ebola virus disease, but there is still no proven method of treating the Ebola virus disease fully. They are still in the experimental stage. Since the Ebola virus disease is still rare in general, there has been little investment given to develop preventive or therapeutic medicines. The experimental drugs that are given to patients are distributed as a clinical trial. This is to help develop better therapeutic agents. With every clinical trait, there comes some cons that trail with it. There are some West African populations that are very suspicious of the Western European doctors. This complicates any attempt in contact tracing, correct burial of deceased cases, and isolation of suspected cases. These complications hinders the use of any experimental drugs in the fight against the Ebola virus disease in the outbreak countries. By involving community leaders in response efforts are needed to alleviate concerns of hesitant and fearful populations. The Federal Drug Administration has brought forward a two-animal rule on drug testing. The two-animal rule states that a drug that has shown efficiency in two different animal models and that has proven not to have any serious side effects in healthy humans can be made available on compassionate grounds. The treatment for Ebola mostly involves rehydration with fluids orally and or intravenous. There are multiple ways that the entire world population can help control the outbreak of the Ebola virus disease. First, if there is a potential case on a patient, health officials can open an active case and give the patient an identification. With this, the patient and or healthcare workers should isolate themselves from the community to prevent the spread of the virus. Secondly, we should identify all the contacts of the ill or deceased. Health officials should be tracking contacts daily for the incubation period, which is 21 days. Constant coordination of the documents when contact tracing that goes into the centralized databases should be a priority for health officials. This is so we can help the families and friends of the patient from spreading the disease further and give them the medical help they need. There could also be an ongoing investigation of retrospective and current cases to document all historic and ongoing chains of virus transmission. Most importantly, the public and healthcare workers should identify all the deceased who have died from the Ebola virus disease, and the families of the deceased should use safe burial practices. Another way is that health officials and the public should be reporting new and updated cases of Ebola patients daily to the Center Disease Control and Prevention. Lastly, affected countries can take precautions by patrolling their borders constantly and enforcing suspected patients into quarantine for the entire 21-day incubation period. If we enhance communication across borders with respect to disease surveillance, we will help control and prevent more cases in the Ebola virus disease outbreaks. There has been some guidelines set for preventing exportation of the Ebola virus to other countries. Authorities should have knowledge of each Ebola virus case and their contacts. which they will not be allowed to leave the country during the 21 incubation period. Travelers should be well informed about the clinical presentation of Ebola, and when travelers are seeking medical help, they should be aware of how important it is to reveal their entire travel history and to indicate any possible contact with sick individuals or wild animals. Following these guidelines, everyone can do their part in protecting themselves and loved ones from contracting the Ebola virus disease. All in all, the world has been on edge on learning how to control the Ebola virus disease that has been attacking Africa since 1976. Ebola does come from a family known as Filiviridae and has 4 other strains than the Zaire strain. Ebola virus disease is known as a zoonotic disease because it can be transmitted from animals to humans. Animals can transmit this virus to humans through blood secretion and bodily fluids. This human disease is thought to result from consumption of poorly cooked infected animals. Some animals that are known to be infected by Ebola are gorillas, chimpanzees, fruit bats, monkeys, forest antelopes, and porcupines. There is evidence that 3 species of fruit bats are maybe acting as a reservoir for spreading the Ebola virus disease. During dry seasons, chimpanzees will eat more fruit bats because fruits are scarce. Humans in the local population will then eat the animals, not knowing the animal is infected with Ebola. That is why educating the local population about Ebola and to properly cooking meat is important and will help stop the spread of this deadly disease. Humans can also transmit the disease to humans easily with any close contact with an infected person. The virus can enter a non-infected person through broken skin or mucous membranes, through blood secretion and any bodily fluids. People can also contract this virus by touching any surfaces and materials that an infected patient's bodily fluids contaminated. That is why healthcare workers are constantly at risk for contracting the Ebola virus disease because they have prolonged contact with infected patients. There is also a risk of contact with an infected patient's bodily fluids or infected needles. Healthcare workers should make sure they understand the importance of the use of personal protective equipment during contact with a patient that is infected and its priorities listed by the World Health Organization. Medical facilities should make sure that the PPE is always available and constantly be training their employees of how to use it. The deceased can also transmit the disease to their loved ones during burial ceremonies if mourners have direct contact with the body of the deceased person. A person is not infectious until they start to show any symptoms of the disease, and they do not transmit the virus during the 21-day incubation period. Once a patient recovers from Ebola, they are no longer infectious. Special investigation can confirm if a patient has Ebola or not. Initially, a patient will have a flu-like symptoms like hemorrhages, like bleeding externally and internally, followed by a transient measles-like skin rash. It is difficult to distinguish EPD from other infectious diseases. After a few days, symptoms start to show. A lab testing should be done either by a polymerase chain reaction or virus isolation by cell culture. These 2 types of testing can be done on the deceased as well. In the lab testing, it will show low white blood cells and platelet counts with elevated liver enzymes. Once a patient recovers from Ebola, for 10 years, the patient develops antibodies that protect them from Ebola. As a side effect, though, the patient might have long-term joint pains and vision problems. Lastly, if the public health officials work together, as we can stop the spread of Ebola altogether. Some ways to stop the spread of this deadly virus is to identify all the contacts of the ill and deceased, use safe burial practices, and report cases daily to the Center for Disease Control and Prevention. The affected counties should be taking precaution by patrolling their borders constantly. They should enforce quarantine for suspected patients for the entire 21-day incubation period. With any type of viral outbreak, panic will arise with the public. That is why evolving community leaders in response efforts are needed to help calm and answer concerns of the public. By enhancing communication across all borders and getting all health officials on the same page, we can control and prevent more Ebola virus disease cases. Furthermore, Once we retain more resources and invest our time and money to finding a vaccine, Ebola can realistically be contained and abolished.